Hi everyone, and you're very welcome to today's Saturday session, which is going to focus on Leaving Cert Biology, and our topic for today is the circulatory system. So throughout today's Saturday session, we're going to look at all the theory behind the circulatory system, and throughout that we're also going to have a look at a mix of exam questions as well, just so we can see the style of question that's normally asked on the circulatory system. Hopefully we'll be able to see a little bit of repetition there as well, just to make sure that we're perfectly as prepared as possible for the final exam next June. So we're going to kick off by looking at the structure of the circulatory system, and the circulatory system consists of three separate parts. So we have our blood vessels, which are composed of the veins, the capillaries and the arteries, and we also have our heart. Okay, so we're going to be having a look at all three of these today in a little bit more detail. We'll start off by having a look at the blood. We'll then start to have a look at the blood vessels and how the blood is transported throughout these blood vessels, and then we'll have a look at the structure of the heart and the stages of heartbeat control. And again, as we look at each one of these, we're going to have a look at a couple of exam questions associated with the three different parts of the circulatory system, just so we're as comfortable as possible with all of them. So the blood contains, we have plasma, we have our red blood cells, we have our white blood cells, and we also have our platelets. Now, if you were to place a sample of blood into a centrifuge and spin it at a really high or really fast pace, it would separate into its individual components. And we can see that in the diagram over here on the right hand side. So the plasma is the straw-coloured liquid. Okay, you can see through it, so it is see-through, and it accounts for about 55% of the component of the blood. So it's the biggest out of the four different components that we have there. Our red blood cells are the second highest, okay, so that's in around 45%. I'll come back to the numbers in a couple of minutes, but the red blood cells, reddish in colour, that's about 45% of the component of blood. And then white blood cells and platelets together will account for less than 1%. So I know there, if you were to add 55% and 45%, you'd hit 100% already, which would leave nothing left over for the white blood cells and the platelets, but these are rounded, okay, that'll be a little bit less than 45%, a little bit less than 55%, and then white blood cells and platelets together are going to be less than 1%. So by far the smallest is the white blood cells and the platelets. So we've already mentioned this about the plasma, but it's the straw-coloured liquid portion of the blood, and it's composed of water, so mostly water. It's also composed of some proteins and also some dissolved materials as well. So the function of the plasma is to transport dissolved materials such as glucose, minerals, salts and hormones around our body. And it also is responsible for transporting some heat, just making sure there's an adequate proportion or distribution of heat throughout our body. It can also transport some heat. Now, we've already mentioned about how it's the liquid part of the blood. It can transport water, contains water, dissolved substances. So we've mentioned glucose and minerals, some proteins there as well, amino acids, carbon dioxide, but it also contains some antibodies as well. So these antibodies are produced by the white blood cells in order to combine with and neutralize foreign substances. And we'll come back to the idea of antibodies in a couple of minutes when we're looking at the white blood cells, but antibodies can also be transported within the plasma. Clotting proteins are also found in there. So we mentioned about how amino acids are there, but a specific type of protein which we can find are clotting proteins, and they're acted upon to form blood clots. Okay, so that's really important if we were to form an injury. Platelets are also involved in this, but it would just make sure that we don't actually bleed out and that there is appropriate clotting within that as well. Can also be a negative side as well, so you might have heard of people with blood clots before and had to get them removed. So both a positive and a negative impact there of clots, but clotting proteins are transported along in the plasma. Now our red blood cells, also known as our erythrocytes, they're red because they contain this substance known as hemoglobin. Okay, and it's that hemoglobin that carries oxygen. Okay, so oxygen will attach due to the hemoglobin, and that can be transported then around our body. Now, they have a very unusual shape. So the shape that they have is known as a biconcave disc. Okay, it doesn't have a nucleus and it doesn't have mitochondria, but the biconcave shape, if you can imagine you had a bit of Play-Doh and you had it kind of as a disc shape and you pinched in the center on either side, that's what a biconcave disc is. Now, why is it shaped the way it is? Well, by pinching that in, you're actually increasing the surface area, and because it's going to carry oxygen, the increased surface area means more oxygen can be carried. Okay, so the biconcave disc increases the surface area, which increases the amount of oxygen which can be carried. Okay, so that's important about their shape. Now, its function, we've already mentioned this, but it contains hemoglobin, which is red in colour, and that carries oxygen. And just a little note there on a deficiency as well, because it is important to know a deficiency associated with each one of these. Anemia is a lack of hemoglobin or a lack of red blood cells, and the symptoms of anemia are pale skin colour and a loss of energy. And that would make sense, because if there isn't oxygen being transported around to all of your cells, you're not going to have as much energy. You're going to find yourself getting a little bit more tired that little bit quicker. White blood cells then are known as leucocytes. So the shape, they're irregular, so they don't have a regular shape. They're not biconcave like the red blood cells. They're irregular shapes. Some of them will be larger than others, and we can see them there. And their function is to fight infection in two ways. Okay, one of them is to engulf and destroy bacteria. That's known as phagocytosis, and we can see that happening in the diagram over here. So we can see this here, a white blood cell. It's a particular type of white blood cell known as a phagocyte. It's going to engulf the bacteria by moving around it. It's eventually going to close fully in around it, and it's going to engulf it inside the cell, where the bacteria can then be digested and broken down, so it's not going to cause us any harm. And the second way that they can fight infection, we've mentioned this already, it can be to produce antibodies to kill the bacteria. Okay, so just to mention there as well, we can see this in the diagram from up here. The white blood cells are larger than the red blood cells, so important to know that as well. And a little note on a deficiency here, leukemia, that's a particular type of cancer in which white blood cells are produced too rapidly and are immature, and then they can be treated with drugs. And the final component we're going to have to look at here is the platelets, which are also known as the thrombocytes. And their function is quite simply just to clot the blood. So if you do have an injury and you start to bleed, your platelets are responsible for making sure that you do clot so that you're not going to bleed out and cause yourself further harm. So hemophiliacs then, they have an inability to form clotting chemicals, and they may suffer from increased blood loss. Okay, so we mentioned hemophilia earlier on in terms of the red blood cells as well, but they do have an inability to form a clotting chemical, and as a result they may suffer increased blood loss because they're not able to clot the blood as efficiently as people who don't have hemophilia. Now overall, we've mentioned this, but the functions of the blood overall, they're going to transport food, waste products and hormones by the plasma, going to transport heat of internal organs by the plasma as well, so it just helps to maintain a constant body temperature. Red blood cells are going to transport oxygen just to make sure that all of our cells are getting an adequate supply of oxygen, making sure that they have enough energy and we're not feeling fatigued, we have enough energy day to day. White blood cells specifically are going to fight in defense against infection, so we have phagocytes and we have lymphocytes, and the platelets then obviously are responsible for clotting the blood. Okay, so if you do suffer an injury, it's going to make sure that we clot and don't bleed too excessively. Now, when we talk about blood, we can also talk about blood groups. Okay, so you've probably heard of this before, people donating blood, but we have four main blood groups. We have A, we have B, we have AB, and we have O. So during a transfusion, so this is when one person is given blood from another person. So during a transfusion, it is critical incoming blood is of the same type as the recipient or from one who is seen as an eligible donor. Okay, so if you have blood type A, now we're just talking generally here, we'll come to the Rhesus factor in a couple of minutes if you're aware of that, but generally if you have blood type A, you will be able to give to somebody else who is also blood type A. If you are blood type B, you will be able to give to somebody that's also blood type B, and if you have blood type AB, you can give to somebody who is blood type AB, and so on. Now following this chart over here though, there is a couple of eligible donors which can step in as well. So if you have blood type A, not only can you give to somebody who is blood type A, you can also give to somebody who is blood type AB. If you have blood type B, you can also give to somebody who is blood type B, but again, you can give to somebody who is AB. And if you are blood type O, you can give to somebody who is blood type O, but you can also give to A, you can give to B, and you can give to AB. Okay, now that would also obviously be really, really useful if that was as straightforward as that, but we will be coming to the Rhesus factor now in a second, which can make a couple of changes to who can give blood to who. But just to remember for now, if you're A, you can give to A, if you're B, you can give to B, AB can give to AB, and O can give to O. And depending on which one you have, you might be able to give to others as well. So O being able to give to A, B, and AB, and then both A and B also being able to give to blood type AB as well. Now, we're going to have a quick talk about the Rhesus factor. So when we talk about the Rhesus factor, Rhesus is just a chemical that's found on the surface of red blood cells. Okay, so we can see over here, this particular blood cell is Rhesus positive because it has the chemical on the outside. This one here over on the right hand side is Rhesus negative because it doesn't have the chemical on the outside. So blood can either be Rhesus positive, which is about 85% of the cases, or Rhesus negative, which is about 15% of the cases in terms of Irish people. Rhesus negative blood can be given to people who have Rhesus positive, but not vice versa. So Rhesus negative can be given to Rhesus positive, but Rhesus positive cannot be given to Rhesus negative because it will recognize that as something that's foreign and your body will try and fight it. Okay, so Rhesus positive cannot be given to Rhesus negative, but Rhesus negative can be given to Rhesus positive. So if we were to look at the chart that we had a couple of minutes ago, I'll just quickly do out another one here. So you can have O positive or O minus, A positive or minus, B positive or minus. Now this isn't going to hold true for everybody. I'm just going to use it to help explain. Okay, so if you had blood type O positive, you can give to somebody who is blood type O positive, but you can also give to somebody who is A positive, AB positive, and B positive. Okay, you can't give to anybody that's negative because the body will not accept it. However, if it was O negative is seen as the universal donor. Because Rhesus negative blood can be given to anybody. So not only can O negative be given to O negative, it can also be given to O positive. It can be given to both A positive and A negative, both B positive and B negative, and AB positive and AB negative. So that's why it's known as the universal donor. You can give to everybody. Okay, so if you've negative, you can give to positive and negative, but if you've Rhesus positive, you can only give to positive. So it just complicates who can get blood from who, and it is important to know what blood type you are, just in case you might ever need a blood transfusion. Now, for higher level, we need to go into a discussion of the different types of white blood cells as well. So we can see here we have two different types. We have our lymphocyte and we have our monocyte. How do we distinguish from each one? Well, we can see here in the monocyte, they're much larger and the nucleus is much larger. The lymphocyte is that little bit smaller and the nucleus is a little bit smaller as well. Now, what do lymphocytes actually do? Well, they comprise of about 25% of all white blood cells, so about a quarter. They're made in the bone marrow, like lots of blood cells, but they mature in the lymphatic tissue. So the lymph system, or the lymphatic system, is primarily responsible for fighting infection. It's our human defense system. So they mature in the lymphatic tissue, and they're stored in parts of the lymphatic system as well. Okay, they're going to be responsible for fighting infection, so we're going to associate them with the lymphatic system. So the spleen and the lymph nodes is generally where they will be stored. And their function, the lymphocytes, is to produce antibodies, which are substances produced by the immune system in response to foreign substances. Okay, so the antibodies are going to help us fight infection within our bodies. The monocytes then, they comprise of about 5% of white blood cells. Again, they're made in the bone marrow. They have a kidney-shaped nucleus, so that was the larger nucleus we seen a couple of minutes ago, and they will generally only survive for between 6 and 9 days. So we seen earlier on the process of phagocytosis, and that's where bacteria is engulfed and digested. It's the monocytes that are going to be responsible for that. So we're going to have a quick look at an exam question here then. So this one was all to do with blood cells, and the first question there says, what word is used to describe the shape of the two faces of these cells? So it's going to be biconcave. They're biconcave discs. If you wanted to talk about one singular face, that would be concave. Because there's two of them, it's biconcave. Okay, so either one would be accepted there. Red blood cells are wider than some capillaries. What feature of red blood cells allow them to pass through the narrow capillaries? Well, they're flexible. Okay, they don't have a nucleus or a mitochondria inside, so they are that little bit more flexible so that they can pass through the narrow capillaries. So it doesn't matter if they're squeezed in a little bit, they're still going to be able to fit through. So name the molecule in red blood cells that carries oxygen. So that's our hemoglobin, which we seen a couple of minutes ago. Human red blood cells live for about 120 days. Give a reason for this based on the composition of these cells. Well, as I said there a couple of minutes ago, they don't have a mitochondria. Okay, so there isn't going to be a huge amount of energy there for them. So that's why about 120 days is going to be the max. Give a location in the body where red blood cells are produced. So we mentioned that most of them are produced within the bone marrow. And where are they broken down? That could be the liver or the spleen. Okay, red blood cells are transported in the blood plasma. Name the transport fluid in humans that does not contain red blood cells. Okay, so we haven't fully looked at this, but we did mention the lymphatic system that can be transported around in lymph. Okay, it's the fluid as part of the lymphatic system, which does go hand in hand with the circulatory system, but we're not going to have a massive look at that today. We'll have a look at that in another Saturday session. But for today, if you just focus in on parts A, B, C, D, and E, that's more than enough for today, and we'll have a look at the lymphatic system at a later point. So we'll have a look at another exam question here. So state 2 ways other than colour in which red blood cells differ from white blood cells. So this is B part 1. So one way is red blood cells are smaller. Okay, and another reason there, red blood cells don't have a nucleus. And we'll just make sure we also mention white blood cells in our answers here, just to make sure we're definitely sure we're going to get the marks. So red blood cells are smaller, white blood cells are larger, and red blood cells don't have a nucleus, white blood cells do. Okay, there's a couple more you could mention there as well. I know we were only asked for 2 there, but you could say about the shape. So red blood cells have a regular biconcave shape. White blood cells have an irregular shape. Okay, so there's loads there that you can choose from. Part 2, name a group of white blood cells other than lymphocytes. So one of these you could have said the phagocytes. So we've had a look at these already. You could have also said monocytes there as well. Okay, and part 3, lymphocytes may be divided into B cells and T cells. B cells produce antibodies. What is the role of antibodies in the body? So they are to inactivate antigens. Okay, so we've had a look at that already. Okay, we can also say fight infection there. So we've had a look at that. Now we haven't explicitly looked at the types of T cells just yet. Again, that's going to come into more in the human defense system. But we are just going to mention what the types of them are, just for if you're having a look, you might recognize it yourself. But the types you could mention there are the helper, the memory, the killer, and the suppressor. Okay, and then you'll be able to give roles depending on each one of those. Okay, so we're just going to mention them now, but just so you're aware, part 1 and 2 and the first part there are the most important ones for us. We'll come back to the other ones when we look at the human defense system in another Saturday session. We're now going to have a look at the structure of the heart and the blood systems that exist. So there's 2 types of circulatory systems that can exist. We have an open circulatory system, or we can have a closed circulatory system. So an open circulatory system is where blood leaves the blood vessels, enters animal cells, and then re-enters the blood vessels. Okay? The closed circulatory system is where it remains in a continuous system of blood vessels. So an example of the open circulatory system will be crabs and spiders, where we're an example of a closed circulatory system because our blood doesn't actually leave the blood vessels. Okay, so the open circulatory system, it actually leaves the blood vessels, enters the animal cells, and then re-enters the blood vessels, whereas the closed, it just remains in a continuous system of blood vessels, your veins, your arteries, your capillaries, which is exactly what happens in humans. So we're an example of a closed circulatory system. So here's a diagram of an open circulatory system. So blood will leave the blood vessels when entering animal cells. We can see that happening down here. So the blood will be passed through the blood vessels, it will leave into the animal cells and then come back into the blood vessels and continue to travel around. The closed circulatory system, though, we can see it's a continuous movement around within the blood vessels for the blood. So it doesn't actually leave the blood vessels and go into the animal cells, it keeps within the blood vessels, and anything that might need to leave from the blood vessels can be diffused out like oxygen. Now, what does the circulatory system look like in its composition? So we have the blood, which we've already looked at. We have the heart, which we'll start to look at in a couple of minutes. So this is what's responsible for pumping blood around the body. And we also have our blood vessels as well. and these blood vessels are responsible for carrying blood to and away from the heart. We'll have a look at the blood vessels first of all. So we have three main blood vessels. We have our arteries, we have our veins, and we have our capillaries. So the arteries, if we look, we have A for artery, we have A for away. So arteries carry blood away from the heart. Veins carry blood to the heart, but because they have the letter V, they also have these structures called valves. So that's how we remember that veins have valves, arteries don't have valves. And the capillaries are these ones here in the centre, which are joining our arteries and our veins together. So arteries bring blood away from the heart, veins bring blood to the heart. They do have structures called valves as well. We'll figure out why in the next couple of minutes. And capillaries are much larger. They're usually actually just one cell thick. We'll see why in a couple of minutes again, but it does help with getting things into and out of the blood because the blood itself doesn't actually leave the blood vessels. So just a quick comparison here of arteries and veins. Arteries will have a thick elastic wall, veins will have a thin elastic wall. Arteries do not have valves, whereas veins do have valves. Okay, so V for valve, V for vein. The reason that arteries don't have valves is because the blood is pumped under much higher pressure. Because it's pumped under lower pressure in veins, there is a risk of backflow. So you don't want the blood to flow backways in the blood vessels. So that's why they have valves to prevent that. There's no worry about that in arteries, so they don't need to have valves because the blood pressure is so high, it's been pumped at a much higher pressure and a much higher rate. Because we have a thick elastic wall here in the artery, we're going to have a narrow lumen. So the lumen is kind of like the hole in the centre, okay, where the blood will pass through. Because the elastic wall is much thicker, the lumen is going to be narrower. But whereas in veins we have a thinner elastic wall, we're going to have a much wider lumen. And we'll see a diagram of that now in the next couple of minutes as well. And we've already mentioned this about how our arteries carry blood away from the heart and veins carry blood to the heart. So we'll have a look at a diagram here then of arteries and veins. So we can see the elastic wall in the artery is much thicker, which means the lumen, which is here at the centre, is much narrower. The elastic wall in the vein is much narrower, so the lumen is much wider. So the walls are both very similar, it's just that the veins will have a much thinner elastic wall compared to the artery. So again, the veins are also going to have these structures called valves. Reason being, because the blood is going to be pumped under such a high pressure in here in the artery, there's no chance of it flowing backways. Whereas because it's under a much lower pressure here in the vein, it has to have valves to prevent any backflow. So again, just a quick diagram of arteries and veins here. Looks a little bit different here, but we can see the elastic wall is prominent here in the artery, not as prominent here in the vein, which means the lumen is a lot smaller in the artery, but a lot larger in the vein. And just to have an idea of what the valves actually look like, we can see they're not present down here in the artery, but they're these structures here we can see in the vein. So the blood can pass through the vein, but there's no chance of the blood actually coming backways, because once the blood passes through, the valve will shut off to prevent the blood coming back the opposite way, just to prevent any backflow, which could be potentially dangerous for humans. Our capillaries are responsible for connecting arteries to veins. They have very thin walls, so it's a layer of endothelium cells, and they allow for the exchange of materials between blood tissues. So when we mentioned there about having very thin walls, they're usually just one cell thick, which allows for the easy passage of, say, oxygen by diffusion or anything else that might need to get into or out of the cell in a similar process. Okay, so really thin walls, usually just one cell thick, and allows for the exchange of material between the blood tissues. The heart then, this is located between the lungs, slightly more on the left side as you're looking down, and above the diaphragm. So made of cardiac muscle as well as a double membrane called pericardium. And the cardiac muscle that's present within the heart is composed of contractile tissue, and it's very slow to fatigue, okay? It doesn't tire. So your heart beats involuntary, you don't control it, and it's the cardiac muscle that's responsible for it. Okay, so really important that we're able to draw the structure of the heart. Some of these you might need to be able to label. I'll go through all of them in the next couple of seconds, but you do need to be able to draw and label the heart. It is a very common exam question which comes up for you to be able to sketch it or for you to be able to pick out some individual features within it. Okay, so what do we actually need to be able to do? Well, we have 4 blood vessels up here at the top of the heart. We have our vena cava, which is a vein because it's bringing blood to the heart, not away, it's a vein, so the vena cava. The blood is going to come in and it's going to exit through this artery up here, which is the pulmonary artery. Now, why is it called the pulmonary artery? Well, up here, we're going to have our lungs, and anything associated with the lungs gets this term pulmonary. Now, it's going to the lungs because at the minute it doesn't have any oxygen. It needs to fill itself with oxygen. It's only going to get that within the lungs, okay? So the reason it's called pulmonary artery is because it's bringing blood away from the heart, so away, or A for artery, A for away, and it's going to the lungs, so it's the pulmonary artery. Now, once it goes up here to the lungs, it's going to start to bring the blood, which is now filled with oxygen, back to the heart. So again, it's coming from the lungs, so that's why it's getting the pulmonary term, and it's coming to the heart, so it's a vein, pulmonary vein. The blood is going to come in, and it's going to exit to be pumped all around the body, so it's leaving through an artery known as the aorta. Now, if we have a look at a couple of the labels here, you might have noticed this already, but if we have a look at the right-hand side, so over this side, the labels have the idea of left. Over on the other side, it has right. The reason for this is when we're labeling the heart, we have to imagine we're looking down at our own heart. Okay, so the left will be over here as we look down at our own, the right will be over here. Now, we have 4 chambers we're going to look at. We have 2 chambers up here at the top. They're known as the atria. So we have the left atrium and the right atrium. Okay, that's the 2 atrium there. And then down at the bottom, we have chambers known as ventricles. So we have the left ventricle over here, we have the right ventricle over here. Okay, so we're left and right atrium, left and right ventricle. Now, we do have some valves in there as well. So over here on the right-hand side, we have our tricuspid valve. So there's an or in tri for tricuspid valve. We have our bicuspid valve over here on the left-hand side. Again, left-hand side as we look down. And then up here, in our pulmonary artery and in our aorta, we have what's known as our semilunar valves. Okay, they're just going to stop any blood from flowing back into our heart. So there are semilunar valves, literally just there at the exit of the heart. Okay, you can see both of them up there. So again, really important that you're able to draw and label the structure of the heart, because you will be asked that as a very common exam question. Now, we mentioned already about the valves. So normally the veins have the valves, but the heart has some valves as well. So over on the right-hand side, okay, the left, as we were looking at the diagram the last slide, but you have to imagine you're looking down at your own heart. On the right side, or for right, we have our tricuspid valve. On the left side, we have our bicuspid valve. Now, we also have our semilunar valves. Just actually, I mentioned these as well. Our tricuspid and bicuspid valve, they're located between the atria and the ventricles. The semilunar valves, they're located directly up at the top of the heart. So they allow blood flow out of the heart and into the main arteries, but prevent the blood from getting back into the heart. Okay, so they're located right up at the top of the heart, just where the blood is leaving in both of the arteries. The tricuspid and bicuspid valve, they're located between the atrium and the ventricle. Now, we briefly touched on this already about how the blood has to go up to the lungs to get oxygen to be sent around all the body, but we can see we can actually split the heart into 2 circuits. We have our pulmonary circuit and we have our systemic circuit. So the pulmonary circuit, again, pulmonary being anything to do with the lungs, is going to deal with the portion of the blood flowing around within our circuit, within our circulatory system, that goes to the lungs. The systemic circuit is going to deal with the portion that deals with the rest of the body. Okay, so if we have a look for deoxygenated blood, this is any blood that's existing there in blue. We can see it's shaded in blue. This is normally the colour that's used to show blood that's high in carbon dioxide and low in oxygen. We can see here that the deoxygenated blood is going to come back into the heart. It's going to be pumped right out of the heart up to the lungs, and it's going to fill up with oxygen once again. Okay, so it's going to fill up, be rich in an oxygen supply, come back into the heart, into the atrium, down into the ventricle, and eventually be pumped right around the body to all of the cells that need it. And once it's used up, that blood supply is going to be high in carbon dioxide again and low in oxygen. And the exact same process will happen again. So once the oxygen is used up, we will have deoxygenated blood. We'll come back into the atrium over here on the right-hand side. Again, it's the left-hand side as we're looking at it, but we call it the right atrium. We're going to come into the right atrium, down into the right ventricle, be pumped out up into the lungs, where it's going to pick up an oxygen supply. It's going to become oxygenated blood because it's high in oxygen, down into the left atrium, the left ventricle, and then be pumped out again to be used up by all of the cells in the body. Okay, so what actually happens? So the deoxygenated blood, which is blood low in oxygen, enters the heart via the vena cava, comes into this vein here, exits through the pulmonary artery, up to the lungs, gets its oxygen, comes back down into the heart through the pulmonary vein, and then is pumped out of the aorta to be spread all around the body. Okay, so two different circuits. We've our pulmonary circuit up here, and we've our systemic circuit down here. Over here, the right-hand side, so left-hand side as we're looking at the diagram here, but the right-hand side as we label it, that deals with deoxygenated blood. The left-hand side deals with oxygenated blood. Now, this has come up naturally in the last couple of slides anyway. We're just going to quickly mention here about double circulation. So the heart acts as a double pump, which is separated by the septum. So that's the structure that goes down throughout the centre of the heart. So we've our two circuits, which we've mentioned already. We've our pulmonary circuit, which is the circuit that deals with the lungs, and we've the systemic circuit, which deals with the rest of the body. So the pulmonary circuit is where the deoxygenated blood comes up to the lungs, picks up its oxygen supply, and goes back down full of oxygen back into the heart through the pulmonary vein. And the systemic circuit is where the blood is used or sent throughout the rest of the body. Okay, so when the blood is pumped out through the aorta, it's used up by all of the cells in the body, and it's going to come back into the heart then through the vena cava. Okay, so that's our pulmonary circuit and our systemic circuit. Now, double versus single circulation. The double circuit allows both oxygenated and deoxygenated blood to be kept separate. So we have separate sides of the heart that deals with the deoxygenated blood and the oxygenated blood. And it also ensures that blood pressure is high enough to reach all parts of the body. A single circuit, though, can only produce low blood pressure. And some examples of this would be earthworms and fish. Okay, so the double circuit is very, very beneficial for us as humans. Now, we've mentioned this already, but the pulmonary circuit, where does this actually happen? Well, it starts off in the heart. We have deoxygenated blood. It's going to be pumped up to the lungs where it's going to pick up its oxygen supply and then going to come back down to the heart, oxygenated, full of oxygen. So the blood leaves the heart, picks up the oxygen in the lungs, and it's a very short circuit because if you think about where your heart is in relation to your lungs, it doesn't have to go very far at all. So the right-hand side of the heart, the walls of the right-hand side, are going to be a little bit thinner because it doesn't have to send the blood as far. The systemic circuit then is going to send blood from the heart to all of the rest of the body and then back to the heart again. So the oxygenated blood is going to exit the heart and travel all around the body. The blood is going to lose the oxygen and pick up carbon dioxide once it's used up. And this is a much longer circuit. So the left-hand side of the heart is going to have much thicker walls because it has to pump the blood a much further distance. So we're going to focus on heartbeat now for the next couple of minutes before we finish up. Now, although the heartbeat is controlled by the pacemaker, which is located in the right atrium, we'll come to that in a couple of minutes, the frequency of the electrical impulses can be altered by our brain. Okay, so the pacemaker, known as the SA node, pulses and causes the atria to contract. So we can see our SA node up here in the right atrium. The electrical impulses that are caused from that, from the pacemaker, will stimulate the AV node. So that's the atrioventricular node. Okay, so we have our SA and AV node. So the SA node, sinoatrial node, will pulse, cause the atria to contract. The AV node will then pick that up and it will be stimulated. So the AV node, if we have a look at where that's located, just beside the septum here, that's going to send the impulses down the septum and that's going to cause the ventricles to contract. So it's the SA and AV nodes that are responsible for keeping our heartbeat controlled. SA node is located up here in the right atrium, the AV node down here at the septum. Now, how can your heartbeat be impacted? Well, exercise, temperature and shock can increase heartbeat rate. Relaxation and sleep can decrease the rate of heartbeat. Now, we've two other keywords here which we're going to come to, and this is diastole and systole, or diastole and systole, depending on what way you want to pronounce it. So diastole is when the heartbeat chambers are relaxed. Systole is when heart chambers are contracting. Okay, so if they're doing nothing, they're just relaxed, it's diastole. Systole is when heart chambers are contracting. Okay, there's a little bit of activity happening in there. Now, how is the heartbeat actually controlled? What actually happens? Well, blood is going to enter the heart. We know that for sure. It enters on both sides, and the chambers are going to be relaxed. So it's allowing the chambers to fill up with blood. Blood is going to be pumped from the atria, so they're the chambers at the top, down into the ventricles. Okay, so because there has to be contractions happening there, we're going to have a period of systole. Okay, atrial systole because the atria are contracting. Blood is then going to leave the heart once the ventricles are full of blood. this is going to be ventricular systole. Okay, so because there was a period of relaxation up here at the top where the blood was entering the heart, the chambers were filling up with blood, that was diastole. Once those atrium or atria were filled with blood, it's going to be contracting, it's going to be pumped down to the ventricles, so that's atrial systole. And then once the ventricles are filled with blood, before the blood is pumped out of the heart, they're going to contract, the ventricles are going to contract, that's ventricular systole. So again, we can see all of that in here. So we have cardiac diastole, so there's a period of relaxation happening there, but once the SA node kicks in, we're going to have a period of systole. Okay, so we're going to have atrial systole first of all. So this is where the atrium up here is going to contract. It's going to pump the blood down here into the ventricle. Now the ventricle is actually relaxing at that point as well. It's allowing it to fill up with blood. So we have ventricular diastole. But once that is filled up with the blood, it too is going to contract so it can be pumped out of the heart. So then the atrium is going to be a period of relaxation, so atrial diastole. But the ventricle is going to have a period of ventricular systole because it's going to contract to make sure that that blood is getting out of the heart. Okay, so diastole is a period of relaxation, systole is a period of contracting. Now finally for today, we're going to have a look at the idea of pulse rate. So your pulse rate is the number of times your heart beats in a minute. The average pulse rate for humans is between 60 and 100, and that changes depending on different animals that you might look at. So in general, it's not a rule, but you might find for the most part, the larger the animal, the lower the pulse rate is going to be, the smaller the animal, the higher the pulse rate is going to be. That's because if you have a large animal like a whale, the heartbeat there per minute could be between maybe 4 and 10 beats per minute because its heart would not be able to sustain pumping blood that far all of the time. Okay, so in general, the bigger the animal, the smaller the pulse rate is going to be. But it's the number of times your heart beats in a minute, the average for humans between 60 and 100. So we're going to have a look at one final exam question here. So it says, draw a diagram to show the structure of the human heart and associated with blood vessels, labelling each of the following. So the vena cava, tricuspid valve, aorta, left ventricle, semilunar valve, and pulmonary artery. So it doesn't have to be an absolute work of art, but we're going to keep it as neat as possible just so we can get all of the structures in. So I'll do it really quickly here. So we're nearly there. Okay, so over here we have our vena cava. That's the blood vessel in there. Over here on the right hand side is where we have our pulmonary vein. The aorta is the artery responsible for pumping around the body, and our pulmonary artery is our blood vessel here. Okay, so we have our vena cava, aorta, pulmonary artery. We didn't actually need the pulmonary vein there. So we have our tricuspid valve. That's located on the right hand side. That's the one between the atrium and the ventricle. So that's our tricuspid valve. Again, I know I'm looking at it. It's the left hand side, but we have to pretend we're looking down at our own heart. So it's on the right hand side. This is the right hand side, tricuspid valve. We need our semilunar valve. So they're located up here at the top, just at the tip of the heart before we get into the two arteries. So we have one there, and we also have one over here. So they're your semilunar valves, and we need our left ventricle. So your left ventricle is down here. So again, it looks like the right hand side as I'm looking at it, but pretend you're looking down at your own heart. So that's your left ventricle. So that's all the labels we needed for that one. State the precise location and describe the role of the SA node. So the SA node is located up here in the right atrium. So I'm just going to write notes on that down here at the side. So SA node found in the right atrium. I'll just write SA node in there as well. And its role is it generates electrical impulses to stimulate a V node. So the AV node then, part 2, do that over here. That's in the septum. And it's near the tricuspid valve. You could also say between the right atrium and the right ventricle, but it's just in here. I'll do it in green. Okay, so that's the AV node. And its function then, it causes ventricular contraction. Okay, ventricular contraction. Now, I'm kind of running out of space here, so I'm just going to do part 3 over here. And it says, what exactly is being measured when a person's blood pressure is taken? Okay, now I know we didn't fully go into blood pressure today, but I will put down the answer here. Anyway, it's the force of blood against the walls of blood vessels. Okay, so we're measuring the pressure that the blood is traveling at. Okay, so there's our label diagram, a couple of questions associated with it. Again, really important that you know that diagram for the final exam. Now, the final thing we're going to briefly mention today is just diseases of the circulatory system. Now, there are a few main diseases of the circulatory system, but the most common is heart disease. Okay, we can see that happening over here, coronary heart disease. We have a healthy artery here, but we can see that there's initial fatty deposits building up inside in the lumen here, up against the elastic wall. This is eventually going to build and build. It's going to stay there and it's going to obstruct blood flow. Okay, so that's heart disease. It's blockage of your arteries. Now we can see that in more detail here. We have normal blood flow happening in the artery here. Plaque is starting to build up here inside of the walls, and eventually it's going to start to increase and form a complete blockage, and a blood clot can then start to form. Obviously they're really dangerous for humans, would have to be removed, so really important that we have a healthy diet and exercise regularly just to make sure that we don't suffer from coronary heart disease. So just before we finish up, I just want to show you around the ExamRevision website. So when you log in, you can see we've arranged a junior cycle and Leaving Cert subjects. All of them are laid out the exact same way. We've video tutorials, we've quizzes, presentations, an exam builder and a resource pack. So if you wanted to find video tutorials based on what we looked at today to talk you through all of the content once again, you would choose the relevant unit. Circulatory System is in 3.2, and we can see there we have loads of videos for you to choose from. So I'll just press on the heart and blood vessels to start off with. And you can see there'll be a video there which will talk you through everything you need to know about the structure of the heart and blood vessels and how blood can flow from the heart through the blood vessels. Now, once you've watched all the videos, you can start to test your knowledge by picking one of the multiple choice quizzes. So we'll pick one there on blood. We can see there's 8 questions. So blood consists of plasma, white and red blood cells, and so we know there the answer is platelets. You can submit your answer, and when you answer all 8, you will get a score out of 8, and you'll find out exactly which ones you got correct and which ones you might have got wrong. Now, when you've done all the quizzes, you might want to access some more notes. So you can go right down here, choose the resource pack, and you will have a full set of H1 notes for all of the topics in Leaving Cert Biology. Now, we just focused on the circulatory system for today. I could easily go back here. That was in Unit 3. Pick Unit 1. If you wanted to find a little bit more about ecology, you can see there's loads of videos there on ecology. Again, there's some quizzes, there's a resource pack full of notes, and we also have our exam builder as well. So you can choose whatever topics you want. All of the past Leaving Cert exam questions are there. The past mock questions are there as well. You can produce your own exam, download the questions, download the marking schemes, and really make sure you know the content inside out and upside down. So if you want to find out a little bit more about it, just go onto our website. You can click on plans and pricing. The more subjects you get, the cheaper it's going to be. It's a monthly subscription. You can also sign up for a free trial as well if you want. If you don't want to commit right now but want to find out a little bit more about it and see how it actually works, that's absolutely fine as well. So just head on to the ExamRevision website, examrevision.ie, and you'll find out everything you need to know. So keep an eye on our social media over the next little while as well. We're going to have Saturday sessions on every week. There'll be a couple more biology ones throughout the year, so keep an eye out for when they're on, and I hope to see you again soon.